I'll send you a video going over that. In fact, if you subscribe to my YouTube channel, you could go through the videos and find the video that describes this. The answers are already posted online. Most of the stuff I do, the content, the whole year's content, most of it's already online, right, on the YouTube channel. So there's really no reason, but I can see if I can find it and then send it to you. If not, I'll make one today during the day sometime and post it. It'll go through the answers that those classic experiments in biology are very important, okay? Because they let us know, and you've started doing your homework, I hope, your chapter homework, because that, I kid you not, you have no time left. You have to keep up with your homework, do as much of it as you can, as early as you can, get your reading done. There's not going to be a lot of time. There's no makeup, there's no resubmissions, right? No late work, because all the work this week is due on Friday. Next week, I'm not accepting anything late. Whatever I don't have by Friday is it. So if you want to retake something, you better schedule it with me during an advisory, because after school every day this week, we're busy, right? And we got a field trip on Thursday. I got ThinkBox every other day. I mean, it's a busy week. Yeah?
All right. Is water wet? No.
This about to be a long.
Say yes or no.
Oh, no. There's nothing long about it. I'm not answering that question. It's nothing to do with what we're talking about. So, yeah.
It's about the thing you gave us yesterday.
If it's a quick question, I'll try to address it.
So when they injected the mouse so the heat killed Smooth and Rough? I forgot. Wasn't that?
Could I have somebody's work? I'll do a short run through it. I'll send you the. Just give me somebody's work. I'll look through it. I remember the assignment. Yeah.
If this strain has died since it can be detected.
Right. So let's talk about that real quick. It's going to be a quick version and the long version, which is going to go through the details. All these experiments will be on the final. All these experiments will be on the final. So no, this you have it. I'm not even going to give you a grade for it. It's yours. You're going to get a quiz on it tomorrow. I'm going to go over it today. Everything in this packet will be on the final. I'll pull some questions right out of these experiments. Are we clear? Each of these experiments is going to have at least one question on the final. All right. So there was a time where we knew these things, DNA, right? And by the way, in the chapter, it clearly goes over what the structure of DNA is. Why would I not go over it here? Because we've already done it. We spent a whole month on biomolecules, and you were tested on it, and you were told you were going to see them again. And now in this chapter in the book, they go over it again. And you have some homework on identifying the difference between DNA and RNA. And that's going to be a real big deal this week and next week. Your final, I will have a study guide for you this week. Traditionally, you have a week. A study guide is due to you a week before the final. You have study guides already. You filled them in. For all the chapters in the book that we've covered, you have a notebook that's full of study guides that I've given you all quarter long. There really is no excuse for, but I'm going to give you a very clear, what I like to call a test blueprint, which is going to break down what this final is going to be about, how many questions, and what form each question is going to be, and what topic is being covered by each question. It's called the test blueprint. So I'm going to go beyond that, beyond the syllabus, then the study guide, and I'm going to give you a test blueprint. All right? But I'm going to wait on that because I need to get it done, and I got a lot going on this week. I have a lot of your work I have to grade. It's over there. That's your Chapter 8 work, all right? And some Chapter 7, some late work from Chapter 7. I'm going to get that graded this week, too. Also, some of you need to retake the test. You have to schedule that retake with me this week during advisory. Am I clear? I'm posting this so that we are all clear and that there's no question that I said it. So you should be writing it down. Now, so they go over what the differences are between DNA and RNA and proteins, but we're going to talk about it here as well. But around the time of 1920s and 30s, we knew there was this thing called RNA. We knew there was this stuff called DNA, and we knew there was this stuff called proteins. We knew what they were made of. We knew that RNA had 4 nucleotides. We knew DNA had 4 nucleotides, and we knew that they were almost identical, except what? That's right. This one had the U, and this one had the T. Those were the kind of the big differences. They both had A, they both had C, and they both had G, right? So they both had A, they both had C, and they both had G. But DNA had the T, and RNA had the U. They knew that. They did. The biochemists figured that out. They identified them. They labeled them. They named them. They knew that there was in a test tube. They could isolate it. They knew it had this test tube. And when they looked at it in this test and when they did their chemical tests, they identified 4 different things and they labeled each one of those 4 different things. That was pretty good work. It took a long time to figure that out. They didn't know what DNA and RNA did, though. They really didn't understand what they did. They knew what proteins were. They knew there were 20 amino acids in living systems. They knew that. And they knew there were there were all kinds of different qualities, right? Some were polar, some were non-polar, some were charged, some were big, some were small. Some had ring structures, some were straight, some were bent. All kinds of different structures, all kinds of different groups. So there was a lot of, right? Which one do you think had the most amount of information? Proteins had a lot of information, right? If you're just looking at information, you're like, this one had the most information in them, where these had kind of standard, right? These two were kind of the same, right? Maybe a little different because one had a U, the other a T. But you only had 4. If you were going to build a book of life, a book that defined every part, or at least structurally defined every part of you, right? Something that was like, I need to send this. This is the book that I'm going to send to my children and my children's children from now until the end of time. This information, I'm going to send that information somewhere. In which one of these 3 would you use as your alphabet? The one with 4 letters, the other one with 4 letters except it has a T, or the one with 20 letters? The protein. I would use the protein. And most scientists at the time thought that proteins was the inheritable material. Proteins were where the book of life was written, right? That all your genes were in proteins. That's what they thought, and it made sense, right? It made a lot of sense. But it turned out it was DNA. DNA was the stuff that held all the inheritance. The genes were in the DNA. Those things that Mendel talked about that we'll cover when we get back from our winter break. That stuff that Mendel talked about, those little inheritable things that the peas sent that made the peas tall or yellow or whatever that you covered in middle school. All that stuff, it turns out, was DNA. Passed on from one person to the next, from one parent to the next parent, from one flower, one plant to the next plant in the lineage. And it turns out that proteins were not the stuff of life. But how did they figure that out? How did we figure out that it wasn't the proteins, that it was the DNA? How did that happen? And that's really an important experiment, isn't it? Isn't it important that you know how we know that DNA is the stuff of life, the stuff that sends information from one generation to the next? So what they did in the experiment, right? And who remembers what they did in the experiment?
They stuffed the mouses with they injected the mouses with that
Bacteria. Okay. What did they do with the bacteria then?
they injected it in
They put it in dishes.
Put it in dishes and put the mouse on them. Some died, some stayed alive.
That's right. That's right.
I didn't get that one, an unexpected one. I didn't get that one because it was, like when they added both the bacteria together, one strain,
the thing died.
Right. Right.
one strain.
Okay. Yeah. It's confusing. I hear you. It's confusing. It's not an easy. It's actually an elegant experiment. It's a beautiful experiment. The results are clear, and you get real quick results. Sad, a mouse dies, right? But it's very clear what the results are. So you have this bacteria. It's a pneumococcus. What do you think pneumococcus do? They produce what? Pneumonia. They produce pneumonia. And so that's one way you can get pneumonia. Another is a viral pneumonia, but it's a bacteria. So bacteria, is a cell, right? So we know it's a cell. We know, you should know that by now. It's going to be on the final. So you see how I'm saying these things? If I were you, if you're a student in college and a professor says that, you're writing that down. So you have a bacteria cell. You know it has kind of a little DNA thing. There's no organelles that have membranes inside. There's protein. There's little ribosomes. There's a little thing. There's little plasmids. That kind of thing, right? So it's a bacteria, but it's not this shape. It's not oval. In fact, the word coccus means it's circular, so it looks more like this little circle, little things like that, right? And there's two kinds. There's this smooth kind, and then there's this rough kind. All right, so I'm gonna just put little red dots on it. And it turns out that the smooth kind was the deadly kind. Do you remember that? The smooth kind was a deadly kind. They did experiments. They say, okay, if I put the rough kind into the mouse, then the smooth kind kills it. If I put the rough kind in the mouse, then the mouse doesn't die. And the reason for that is these things here, and just so you know, and I explained it in the paper, okay? These that you read, hopefully you read it. These little rough dots on the outside, this is a cell, right? That's a cell membrane. What are the rough dots? What makes it rough? What are the dots? No, they're on the membrane. Ribosomes aren't found in the membrane, the plasma membrane. What's found in the plasma membrane? Good guess. The kind of enzymes are proteins. They're integral membrane proteins, right? That's what you find in a membrane. Phospholipids and proteins. You should know that by now, right? Phospholipids and proteins are in the membrane of the cell. If you have these proteins, these red proteins, well, the mouse's immune system can identify them. So when you have these proteins, it turns out that these often proteins that are on the outside of the cell are how you identify who you are. When we go over genetics, and that's going to be one of your winter break packets, okay? He's going to be going over some of the stuff. Yeah, you have like 3. Yeah, you have like. It's 3 weeks. You have 3 weeks off, 1 a week, or you can do all 3 of them quick. We can spend our class time doing this if you'd like, talking about this rather than going over the content. But that's up to you. About how many packets we have over winter break. But we have, you're not even listening to yourselves, to me. But in any case, you know that there are 3 blood groups, right? There's A, it's actually 4, B, O, and then there's AB. And we'll talk more about this, but I need you to understand that the reason, if you have A blood type, the reason you have A is because on the surface of your proteins you have these kind of, I'm making them into triangles, but you have these little green triangle proteins on the outside. And so when your cell sees these cells with green triangles, what does your body do? Nothing, because they say, hey, this is me. I'm okay. This is part of me. I'm good. I'm cool. It's like in the old days when I was a kid, you had the Bloods and the Crips, and they had their colors.
We still got.
I mean, not like they were when I was a kid. When I was a kid, they were all out. Everybody was everywhere. But today, but your cells are the same way. They go and they identify, are you part of me or are you not part of me? If you're not part of me, they attack. They kill it. So if I take a pencil and I stab you with it and break the tip off in your body's going to go to it. Your cells, your white blood cells are going to go and try to sense it. Once they sense it's not part of you, they attack it. They kill it. They try to kill it. It's dead already. But they try to kill it, and they'll push it. Literally, it happened to my brother when he got a splinter. Over time, your area will get real red, and it'll literally push it right out of your body. Almost like in a movie. Of course, it takes weeks to do it. But it's called, you might even get an infection, but the infection is when these cells multiply faster and your body can kill them, right? So if you're a B blood type person, then you have these, you have these kind of, let's call them circles on the outside. So the A is triangles, B is circles. O blood type, you don't have any proteins on it. I'm an O blood type, and we don't have any proteins. We don't have any of these particular proteins, right? And because of that, we're called the universal donor. So we could give our blood to anyone because we don't have those proteins on the outside of the cell. So no cell is going to read it and say, oh, this isn't me. They're going to read it and say, well, I can't believe you're on your phone, dear. I cannot believe you're on your phone. Your notebook is not open.
It is open. People tell you do it, you really got to learn.
Okay, you need to leave. Go outside my room. Take your stuff. Go outside the room. Go to the office. Do what you got to do. Don't come back with a jean jacket on. And when you come back when you're ready to learn with a notebook open and not your phone in your hand. Thank you. Yes.
Is there any blood type that is?
Say that again.
Is there like a blood type?
Well, let me get to the end. Let me get to the end before you ask another question. So B blood type has these, I'm going to call them circles. They're not circles, right? And they're not triangles, and they're not green, and they're not blue, but they're proteins. They're on the outside. O blood type doesn't have these proteins on the outside. So when a white blood cell, if I give you my blood, your body looks at my blood cells and doesn't see any proteins on the outside, so they're cool with it. There's no identifier. So again, I like the gang analogy because it's like if you're wearing red and you go into a Crips neighborhood, you're going to get in trouble, right? Or if you're wearing blue and you're going into a Bloods neighborhood, you're going to get in trouble, right? Because they're going to identify you as the opposite, as not being part of that particular group. Well, your red and your blood cells are the same way. So anytime you give an A gives blood to an A, everything's cool because they're in the same group. They have the same identifiers. B is the same thing. Those are cell surface proteins, right? B has the same. It can give to B, but it can't give to A because A will say, "Whoa, no," right? O doesn't have any identifiers, and so O can give to anybody. But you know what? O cannot take from A or B, right? Because if you put A blood type in me, my body's going to say, this is not me, attack. If you put B blood type in me, my body says, that's not me, attack. So I can only take blood from people with O blood type. Wait, 1 second. Now, people with AB blood type, and this is why we discussed this topic in genetics, they express two different genes. Obviously, there's a gene for A, there's a gene for B, there's a gene for AB, okay? People with AB blood type, they have both triangles and circles. So they have a parent who has A blood type and a parent who has B blood type, and these genes are both expressed. So this is not a dominant recessive situation. This is what we call codominance, and we'll talk about that after winter break. And in codominant situations, you see both genes showing up. So both sets of proteins are expressed. So people with AB blood type can take blood from A. They're called universal acceptors because they could take blood from A. Why? Because they have the A identifier. So their bodies can't tell that A is not part of them. the B, they could take blood from B because they have the B identifier. They could take blood from O because O doesn't have any identifiers. So they're called the universal acceptor. They can't give to anybody except AB.
So can you use it in
I'm not sure how to use that and identifier, except that it would be like if you walked into a Cribs neighborhood and you were wearing a blue hat, then you kept walking. You're going into a blood neighborhood, so you'd switch hats to a red hat. So, or if you rather think of it, you have a hat that's one half's red, one half's blue, right? So other than, and then if you're an O, you don't have any hat at all. Does that make sense?
Yeah. Is there such thing as O negative?
Yeah, we'll talk. That's another story, but it has to do with a smaller protein that's also an identifier. But so the people that have A blood type have this genotype. They have a double A genotype. People that have B blood type, they have a B genotype, and people with O have what are called they have both genes are broken. They don't have A or B. All right. Where AB obviously has AB genotype. Does that make sense to everybody? So that's how cells work. Well, it turns out that this cell here, these two cells, the smooth strain has no cell surface protein. So and the rough strain has these red ones. Well, when you take and inject this red one to a mouse, what does that mouse do? It kills it. The mouse says, hey, wait a minute, that's not me. It's dead. I'm going to have to kill it. And the mouse's immune system kills that bacteria. But you take this smooth strain and you inject it into the mouse, it dies because there's no cell surface protein to tell it what's going on. How did it get those proteins, though? That was the question. This is, by the way, how we know that DNA is the inheritable material for life. So the way that they did it in the experiment, again, very beautiful experiment. I need you also to know that control groups are going to be heavily tested on the final. You guys did horribly on your essays overall, identifying what was the control group or what was the experimental group. In fact, if you did less than 3, if you scored it, there were 5 possible points. If you scored less than 3, I didn't even put the grade in your grade book. I left it out. So if you scored a 3, 4, or 5, then you got your points in the grade book for the essay. So it's not that I didn't grade them. It's that you guys obviously have demonstrated you still don't understand what a control group is. Some of you are having trouble reading a graph. You're still having trouble reading a graph. It's a real problem. So if you don't see a grade in that essay part of that test, you need to see me after some time to really have a deep discussion over how to read a graph and what is a control group and what's an experimental group. We're not going to do it here, though. All right, so you have the smooth and the rough. Those were the two. So what the experimenters asked was, what's being inherited? How come they had an idea why the smooth and rough, what the difference was in their lethality. And so, of course, what they did is they took the rough strain, they injected it, the mouse lived. They injected the smooth strain, the mouse died. They killed the bacteria with heat. They killed the bacteria. They injected that into the mouse, and it lived. Why would they do that? Why would they kill the bacteria by heating it and then inject the dead material into the mouse?
To see if you would have some type of reaction. I mean, to see.
We call that a control group. A control group is something we're using to compare our results to. So we have these mice, and there's 3 of them, right? In fact, let me do something else rather than trying to draw them. you have these mice. This is a control group. So you have these mice, and I know they're horribly drawn, deal with it. And you have 3 of them, or actually 4. To one, we're doing an experiment, right? We want to see what's being inherited. We take and we put the rough strain RS in there. The mouse lives. There's 3 people talking. I'm not going to mention names, but when I have a parent conference and they ask me why you're not doing well, I'm going to say, let me refer you to this video. Let me tell you that the person I'm talking to in this video was your daughter. And you need to understand that if she's talking in class, she can't understand what's going on. Reasonable conversation, yes?
Yeah.
So rough strain goes in there, the mouse lives. What does that tell you? That they're not deadly, right? And then I inject the smooth strain. Even if you don't know about cell surface proteins, you know the smooth strain dies, right? It kills the rat, the mouse, whatever. Then you know that then there's something about that smooth strain that makes it deadly, right? But then I take and I do a control, because what were these? What were the rough and smooth? They were also controls, right? Because I knew the smooth killed, and I knew the rough didn't. But why did I inject mice? Why did I even inject a mouse to kill the mouse if I knew it was going to kill it? I need to have something to compare it to. So these 3 are controls. This is what I call my positive control. This is called the negative control. Why is this positive control? Because if I'm saying the smooth bacteria have this deadly trait, then I have to prove that they're deadly, and I have to show that this kills mice. And this is it. This is what I'm going to use to compare it to. And then if I think that the rough smooth doesn't, I have to show that. That's a control group. So now I kill them. I kill the bacteria, and I inject those. And guess what happens? It lives. And what does that tell you? That means the bacteria have to be alive. So so then I take the heat kill. I'm going to go HK if you don't mind. And I take the smooth strain. I'm going to go at SS, smooth strain. Oh, no, sorry. Smooth strain heat killed, right? So I should say that. These are both smooth strain. I take the rough strain, and plus the heat killed, I inject them, and guess what happens? I let them sit in the it dies. What does that tell you? What did that experiment just say? By the way, this is your experimental group. This is the one you did not know what was going on. You couldn't predict it. You knew the smooth was going to kill them. You just did it to use a comparison group to make sure that your smooth strain were actually deadly and your rough strain were not. You wanted to verify that. So that's why you did these 2 control groups, the positive and the negative. And you heat killed one. It was another control group that you had to make sure that you could show heat killed by itself doesn't kill anything. Does that make sense? But now I mix the heat killed with the rough strain. I inject it. Now it dies. What do you think happened? What's your hypothesis? Say again loudly. If the bacteria died, what's killing the mouse? It's not a toxin. How do I know it's not a toxin? Because my control group, my heat killed control group, right? These are killed. I inject everything in that test tube into the mouse, and guess what? So I know there's nothing poisonous in there. That's why I have this control group, to show that it's not something, I'm not injecting these things with some kind of poison. That's why I do this control group, because I got to show that there's no poison in the sample. So now I take these heat killed, right, and I mix them with rough strain, and then all of a sudden it kills them. Kills the mouse.
So I guess
Not too much bacteria, but good guess. The same amount was put in all the test tubes. Yeah. But it's a good guess. That's one of those factors that you would control, right? You keep the same, because you're only changing one thing, that you're mixing the heat killed with the rough strain. Yeah?
So when the heat killed, because in the third one it said the heat killed when it was injected into the mice lived because it was already dead. So if they injected the dead one and the mice was able to live, the rough one wouldn't the immune system be able to detect that one, so it's still...
Right, so that's right. See, oh my God, that's exactly the kind of thing you got to be doing. It's perfect thinking. You have your rough strain is going in there. So according to our experiments, the mouse's immune system should be able to identify this and kill it. We know the heat killed is not killing the mouse. Why? Because we have this one that says it lived. But something's happening, right, that makes this combination deadly. What do you think is happening? They changed. What changed? Right, the live strain must have changed. So what does that mean? That means that somehow this bacteria that's rough, right? This rough bacteria somehow got the what? Some kind of inheritable material from the dead one, right? There's these pieces, they're dead, right? There's just all these pieces. Some of this material here must have gotten into this cell and changed it, right? So it changed it. It's the only thing that they could think of. So somehow this bacteria changed from rough to what? Smooth. To smooth strain. Somehow some genetic material changed the rough to smooth. So what was that genetic material was the question. So then came the second part of the experiment, right? So here's what they did. They said, okay, well, what if we have these enzymes. Enzymes, remember, they speed up reactions. Well, RNase, what does RNase do?
It breaks down the DNA.
Excellent, 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 excellent. I love that you're thinking like that. It means you did your homework, you did your classwork, but you got a little bit wrong. It's not DNA, it's what? Look at it carefully. RNA. It breaks down the RNA. So it takes RNA and it breaks it down into nucleotides. So no more RNA. No more. So you get rid of the RNA. It's breaking it down. Protease does what? It breaks down proteins. That's right. No more proteins. And DNA, do you see how beautiful this is? Because now you have some tools that let you get rid of DNA, or get rid of protein, or get rid of RNA. So they took this, so what they did, then these are your tools. These are tools. These aren't really experiments, they're tools. So now you take your heat killed, right? Heat killed, rough, smooth strain, right? SS, smooth strain. We know that there's inheritable material. We know that they can pass down that smooth trait to their friends, right? So they take and they put it into 3 test tubes, right? And you should be able to get a lot from this, including the answers that you're going to need for your final. You're going to add RNase. You're going to add to this group protease. And then you're going to add DNase to this group. Oh my goodness, what does that mean? Then you're going to take and you're going to mix this one with what? The rough strain, right? Mix this one, whatever's left over, to the rough strain. And then this one to the with the rough strain. And then you're going to inject it into mice.
Oh, and then you see which one.
Right?
Which one? Oh.
Then you're going to inject it into mice. Yes, I know they're horrible drawings. I don't want to hear it. Say what now? I can't hear you.
Could have just made a circle.
It could be a roller coaster, but it is a mouse. So, like I said, deal it.
showing that the mouse died for the first two?
Yep, that's right. We'll look at it, but you'll see what happens in just a second. So in the first with the first mouse where you got rid of the RNA, hold on a second. You got rid of the RNA in the first one, right? And what happens? The mouse dies. Huh. Could it be that RNA was what passed on the genes for smooth strain to the rough strain? Could that have happened?
Yes.
Could it be that RNA is the stuff that's being sent from the heat-killed bacteria to the rough strain to make them smooth strain? Could it be that?
Yeah.
Yeah, it gotta be.
Yeah.
So you're saying that RNA is making rough smooth?
No, I'm asking you, is it?
I think it got
Possibly.
It could be it could possibly.
Look at this and answer. Think first. Think about it. RNase does what? So I add RNase to the heat killed. I then mix them with the rough strain, and then I inject and the mouse dies. I ask the question, is it possible that RNA is what sent the information to change the heat killed? Or the rough strain? No, why not? There's no more RNA. Excellent. There's no more RNA. It can't be. Look, I love you guys thinking. Look at it. There's no more RNA. No more. Why? Because we added RNase. So this experiment shows it can't be RNA, right? So then I add protease to the sample. And I inject the protease. The protease means it got rid of what? Proteins. And the mouse dies. So it can't be protein. So the inheritable material can't be protein. Man, I thought for sure it was going to be protein. That's what scientists probably thought back then. I can imagine sitting down, reading your journal or the paper at their coffee. These biologists spend their whole life studying this, reading their journal at their morning coffee, their newspaper. They're like, oh, what? It's not protein. I can't believe it. So yeah, it wasn't protein, and that was a shocker. Because remember, it had the most, that's the alphabet that all of us said we would use proteins for the alphabet for the book of life because there's so many letters. But no, that's not it. Hold on to your questions. Then they added DNase. And they had to make sure. They had to make sure there wasn't something else that was sending. Maybe it wasn't any of these. Maybe it was something else that was sending the information to the rough strain to change them. So they added a rough strain, and what happened? They lived. The mice were happy. The scientists were ecstatic. Because what did they just identify?
That was the...
DNA is the inheritable material.
Yep.
So DNA is what makes the mouse
The DNA is what... But remember, here's the tricky part to this experiment. It's a beautiful experiment, but think about it. I didn't say you added DNA. I said you added DNase. DNase gets rid of the DNA.
Right.
And I got rid of the DNA from the heat kill, from the smooth strain, and now the rough strain stayed rough. They did not inherit the material, but only because I got rid of the DNA, proving beyond a reasonable doubt that DNA was the inheritable material. Now, did I really prove it? No, but I had collected enough evidence to make people confident that DNA was the inheritable material. As it stands, we're pretty sure, after all these years and experiments that we've done, remember this is back in the 20s and 30s and 40s, etc., we're pretty sure that this was definitely, DNA is the inheritable material. Well, it's 9:54. I promise I wouldn't try to take things all the way to the last minute. I think I talked more about this experiment than I really wanted to. But I think tomorrow's discussion needs to happen because tomorrow the Mendelssohn stall experiment is very important in DNA replication. I can tell you that both of these, I've seen both of these in various standardized exams. All right? So I think it's worth our time to have that discussion tomorrow. How do you feel about that? Do you agree? Do you feel more confident now about this experiment? So go back and make sure you understand it. Could you talk to each other about it? I promise you this experiment will be on the test. I promise you there'll be about 2 or 3 questions on the final about this experiment. On this test coming up, on the next test, which will be Monday, not Friday, on that test, it's going to be a big part of that test, but you'll see it again on the final. So work on it during advisory. Come and ask me questions. Catch up. Make sure your work is getting inputted this week into the Jupyter grades so that no one's grades. I want next week to be just us worrying about the final and whatever's left of DNA, RNA, and DNA replication, or transcription, translation, and DNA replication. Are we good? All right.